Okay, good afternoon, everyone. Welcome to the NIH Blueprint Neurotherapeutics Network Q&A webinar. We will begin today with a presentation followed by questions and answers. Please put all of your questions in the chat feature below and they will be answered at the end. This presentation will be recorded and distributed in a couple of days to all who have registered. I will now turn it over to our moderator for the day, Dr. Charles Cywin.
Thank you, Rakonda. My name is Charles Cywin. I'm the Director of Small Molecule Neurotherapeutic Development here at NINDS. I'm going to tell you today about the NIH Blueprint Neurotherapeutics Network. So it is housed in the Division of Translational Research here at the National Institute of Neurological Disorders and Stroke, also known as NINDS. And within the Division of Translational Research, we have a suite of programs that are designed to take you from basic research findings all the way to the first in human type experience. And we do this through different programs. Some programs are at the earliest stage of developing an assay and running screens. Other programs like the Blueprint Neurotherapeutics are taking those screening hits and moving them through the optimization process all the way into the first in human studies. And we have this for different modalities. So I'm going to focus in on the small molecule one today, but do know that, and I may touch on the biologics and devices, FOAs that are out there. So with no further ado, I'm going to move into the Blueprint program at this point. So the Blueprint Therapeutics Network, otherwise known as BPN, it's been in existence for over a decade now, since 2012 in the small molecule space. We have funding opportunities that are milestone-based cooperative agreement programs, and they take you, as I said, all the way from I have my screening hits through the exploratory phase, hit to lead, all the way into the clinic. And in these phase mechanisms, I'll talk a bit more how they work as we go down. We have both a all-comers kind of UG3, UH3, meaning you can be a small business that's not necessarily an SBIR small business. You can be an academic institution. You could have foreign components in there. You can even be a foreign institute. There are additional requirements for foreign institutes. We can talk about a bit further when we get down to that. But the SBIR version of the program is one that you have to be SBIR eligible to utilize. And again, I'll talk a bit more about why you might want to do that as an SBIR program, but there are advantages to this blueprint in terms of resources that are not available through the standard SBIR program. But and so there are differences as well from those programs. Our next application due dates are February of 2024 and July 15th of 2024. So we are making a change. That change is we were previously February and August. We're going to be February and July, and then we're going to move into January next year, and it'll be subsequent years after that. It'll be January and July. Please hold for 1 second. I have to change my microphone battery. Hopefully we're back online. Someone give me a thumbs up, Carolyn. Thank you. Thank you. And so the receipt dates are changing slightly by that month shift, and that allows review more time to get ready prior to council rounds. So what is our vision? Our vision is simply combine the strengths of NIH with industry expertise for small molecule neuroscience drug discovery and development. It all starts with you, investigator-initiated ideas for novel drug targets and strong, disease assays and models. And we combine that information that you bring to us with industry expertise in terms of people, consultants that we've hired that have decades of experience in pharma in different disciplines, as well as we have a suite of contract research organizations that we've contracted with that we can provide you access to. Both of these things are done outside of the grant budget and separate. That's why this is a cooperative agreement mechanism. The entry point for this program can be, as I said, anywhere along the pathway, from exploratory, I just have a screening hit, to hit to lead. I've really done some preliminary SAR. I've shown that the compounds have good properties, and we're ready to start the optimization process in earnest, all the way through lead optimization, drug manufacturing, formulation, pre-development work to IND, tox, GLP tox, scale up for GMP manufacturing, all the way to the clinic. The exit points for this coming in, while the entry is all the way from starting to beginning of toxicology, the exit point can be anywhere along the pathway as well. So it can be as early as hit to lead. We've had a program taken out at the end of hit to lead on more than one occasion, licensed out. And we've also had projects licensed out at the end of phase 1. So anywhere along the line is a good exit strategy for you all to get investments to move these projects forward. The goal of this program is to make sure that the projects that we are funding continue on into further clinical development. We want you to have the tool set in terms of data, in terms of knowledge, in terms of ability to give a pitch by the time you're done with our program, so that you can get investment to continue the funding down the path. The goal itself, it says, is de-risk everything to get you there, identify the best ideas for that translational work. But really importantly, this is non-dilutive funding, right? We do not look for having an intellectual property stake or a stake in your equity in your company. We're looking to preserve all of that. So as such, our consultants assign intellectual property assignments to your institutions, our contract people do manufacturing and intellectual contributions to things like synthesis and analoging and so forth. Again, they're going to assign those intellectual property rights back to your institution so that you have unencumbered IP to go off and do licensing with. The minimum entry criteria, strong premise, small molecule starting point, and SAR driving assays available. That doesn't necessarily mean you have to have in vivo activity to start with. Exploratory projects won't have in vivo. They'll just have potentially the screen and the counter screening data. You may have literature information to justify the preliminary data that justifies the premise. Not necessarily with your particular series, because that's the whole purpose of the program, is to make compounds that are actually good tool compounds for in vivo that we can advance the program further into a clinical candidate. The program itself, as I mentioned, is for small businesses and academics institutions. The grant funding is along the drug discovery pipeline. They can enter as a discovery or development project, and development is, I have a compound, I'm not really doing any more optimization in our world. That means no more med chem optimizations going on. And so you're picking your compound. You may have a few different compounds that you're working on, characterizing, and then trying to get down to, you're really at the end of lead optimization, but we call this a development stage because you're not doing any more medicinal chemistry. And that will take you again through that pipeline. I already talked about what you have access to in terms of the outside people, but the inside people you have access to are people like myself and my team that have years of experience in pharma and drug discovery and development that help facilitate the program and work with your teams and through lead discovery teams that we have on a regular basis. And I'll talk about more about those in a second. We also have science officers that have the entire portfolio of your disease indication in their job and really understand the field quite well and can help make sure that the assays and the things we're doing are aligned with the state of the art. As I mentioned, we have these lead discovery teams. They meet every 2 weeks from the start of the program all the way to the end. They add people as we go along in terms of consultants in there. We have the whole suite of contracts. Curia does our medicinal chemistry work that includes in vitro ADME and computational chemistry. We have PK and tox, both GLP and non-GLP done through a number of vendors. We have drug manufacturing formulation, we have clinical trials. I did not mention, but if you're doing optimization, we'll be using the CDD, Collaborative Drug Discovery Database, a relational database, put the data in from Curio that's generating along with what you're generating into the database. Again, it makes it easier when the time comes for sharing data with potential investors, get it right out in a way that you can share. It also helps when you're coming to write the IND to have things in a handy place. We also have a SharePoint system that we expect everyone to use to exchange data with all the government folks and contractors in a way that preserves your intellectual property. I get this question every time. Do you have to utilize all these resources? No, you don't. We can customize this to your team and make sure that it is aligned with what you need to do. But if you're going to do it yourself, you need to ask yourself the real question is, do you have the expertise and resources to do these activities readily? And I really want to underline the word readily available in-house because this program is a milestone-driven program, it moves rather fast, and it's not realistic to expect you're going to hire a whole bunch of people after you get the grant and still make your milestones because the milestones sort of assume everyone's there on day one, ready to go. And so we can talk more about that, but that is the biggest challenge. So, and of course, since it's milestone driven, if it ends quickly because you missed a milestone, you don't want to have to terminate people on the med chem side, for instance. You can still be involved if you're a med chem person's in there in the design, the elements of it, but you don't have to be the lion's share of the effort of the synthesis work, for instance. Okay, so we have 9 different institutes and centers that are involved in the Blueprint Neurotherapeutic Network, have signed on and are actively willing to fund projects within their mission. And the way it works is the funding for the contracts and the grants are going to be funded by the IC that this mission falls into. So again, when we talk about how you're doing your program, we should always talk about which disease indication you're going for to make sure we put you in the right institute and center to talk with their science officer, make sure they're supportive of what you're proposing. I've already said this a couple of times, it's a 2-phase mechanism, it's milestone driven. There's every project, I get this question every time, can you come directly to the second phase? No, you can't. You must start in the preparatory phase, but the preparatory phase is aligned with the milestones appropriate for where you're entering. So a preparatory phase for an exploratory grant would be, I need to show the assays functioning, have good C prime scores, show that I've benchmarked my compounds and that I have a data-driven plan for SAR. Show me I have figured out my target product profile and that it's a reasonable thing. Whereas if you're coming at the development phase, your preparatory phase might be, I'm running the rat and DRF and dog DRF, or maybe I'm just scaling up the material and running the rat DRF in year 1 to show that I have a therapeutic index on my clinical candidate or potential clinical candidate to get there. So we will adjust it accordingly. What is also important is to recognize that not all of the institutes accept development projects. 2 notable exceptions, NIMH and NIDA, historically have not taken development projects. They're more interested in seeing stuff at early stages. So again, depending on the disease indication area you're aiming for, you may or may not qualify for BPN in that indication area. So we should talk about this. There are certain budget constraints. They're not the same. The SBIR follow SBIR rules. There are some exemptions, but they're by institute. And so what you're seeing here are the numbers for NINDS, but numbers differ for NIDA, for instance, and NIMH. And so we need to talk, if you're having an SBIR, what your budget needs to be. These are direct costs in total dollars. Sorry, these are total dollar costs. These are direct costs. So in SBIR, you're limited to a maximum of $700K in phase 1. That phase 1 could be up to 2 years. Most people, because of the budget limitations, are limiting it to 1 year, which puts you at a 4-year maximum time frame for the SBI RE44 in that scenario, where under the UG3H3 you have a 5-year. This isn't direct cost. You can ask for this much, but if you ask for more than $499,000 in any year of the grant, then you need to talk to us in advance, and you'll have to go through a process to get an ARA process to allow the grant to come in. And so it'll have to be reviewed before it can come in. So we'll have information like a budget justification, some research strategy information in order to understand how the budget relates, and then determine whether we will accept it. And that's true for any grant under any ICs for the UG3 The reality is most people stay under the 500K limit because they're utilizing our contracts, and so it's not much of an issue. I rarely end up with an ARA request. I said I mentioned foreign applications, and I should mention it here that, you know, foreign students are able to apply, but it's a 2-level review. You go through the normal review process just like everyone else. Your application is scored just like everyone else. But there's another set of review criteria that's looked at the end, and then also in our council round, where the reviewers will assess whether the project presents special opportunities for further research programs, such as unusual talent, resource populations, environmental conditions that are not available in the U.S. Readily, or could augment existing resources in the U.S. So you have to make that justification. It has to be robust. Otherwise, it won't get approved by the reviewers or by our council for funding a foreign institution. Entry criteria. I've said this a number of times. Discovery, disease biology. Get your assays up and running as quickly as possible. So certainly no later than 6 months into the program, we expect to have SAR driving assays in place for the in vitro side. You know, if you're entering exploratory, in vivo might be in year 2, so you might have more time to have an in vivo assay up and running in the program. But you have to plan accordingly. If you're coming to hit the lead or lead optimization and you're expecting to be doing in vivo assays, then you need to have those. It's really important to understand that we do have a suite of contracts, but we do not have contracts to do disease biology. We have disease agnostic things like toxicology and PK, but disease biology must be part of your grant. And so the in vitro assays and the in vivo assays for disease biology must be part of your grant budget. That doesn't necessarily mean you can't be through a co-investigator that works with you. It can't be with outside CRO that you've contracted with to do work because you're more of a virtual company. That can work too. Just have to stay within the budgets. From a compound point of view, make sure that, you know, we actually require optimization, and that it's amenable to chemistry, and that it has no IP blocks, intellectual property blocks against it. If you're coming to develop someone else's compound for which you do not own, you would need to show permission for how you're going to do that. From the development side, you need to have a fully optimized compound with strong data linking it to the disease target. You may not have all the therapeutic index done, but you should have a good idea of what the biology of the ADMET looks like to really support the idea that this is a potential clinical candidate. You also should have some input from a clinician that there's a feasible path to the clinic, as well as IP is free of roadblocks here as well. This is up on our website. It's also now part of the funding opportunity. It's linked in funding opportunity as well as discussed in terms of milestones. I got a little more descriptive in this latest version of the funding opportunity to make it clear to investigators in one spot how our milestones are laid out. As I said, they progress through the program. This is sort of our generic set. For your individual program, there may be additional milestones associated with the biology, disease biology. We are expecting the grant to be no more than 5 years in combination, both phases of the program. The phase 1 may continue beyond that year because it's typically done through contract. if it's being done in the grant, then it's beyond the same limitations of the 5-year time frame. Expectation is that you'll follow U.S. patent law, and that means inventorship is followed by, inventorship is inventorship. It's a legal thing. It's not something we choose. Assignment is what you're able to choose. And we've taken care of the assignment part of it, meaning the courier contractor chemist is going to assign that intellectual property to your institution. But they would still be inventors if they are inventors. The program itself is not seeking a stake in your IP. And up front, if you have multiple different moving parts that you're working with, we expect you to have your IP agreements in place, because at the end, we do not want to see you litigating amongst yourselves about who owns what. So having those up front amongst the institutions involved is an important thing prior to award. As all cases, we're trying to get you unencumbered IP so that you can hopefully license this product down the road. We have a great website, and I strongly encourage you to look at it. It has lots and lots of information on it. It has introduction to it, has our funding opportunity links in it, has our current project portfolio. It has a virtual how the pharma model works for us, talks about who our external oversight committee is, all the different resources. In our frequently asked questions are things like the milestone plan, just near the bottom of the frequently asked questions. I think it's the second to last. What other institutes in their context, things I put in today's webinar are also available on there. There's also going to be a link to the webinar on this page as well. So if you get nothing else from today, write this web link down, bookmark it for future reference. So you may ask yourself, been around for a decade, what's the evidence that this program works? And so because we've been around a decade, we have projects that I started in, 12 years ago. I was a project manager on Tetra, this program right here. This is for PDD-4D, and it was advanced as for mild cognitive impairment and Fragile X and Alzheimer's disease. Tetra first licensed it to Shionogi and then sold the whole company to Shionogi. So I'm following what's in their public press releases, and it presently shows that it's in phase 3. Likewise, Polite started as an academic group. This was a small business. This is an academic group out of Columbia. They licensed the program at the end of phase 1 to Polite, and it is now in phase 3 for both Stargardt's and dry AMD. We have a program we started and for one indication, was bought by a company for another indication. They progressed one compound that we created into that, and we continued for migraine, and eventually they were sold again. Migraine was benched at that point, but the MDD project still continues, and again, I see it's in phase 3. I'll point out, I haven't updated this slide. I just saw information recently that this program finished their phase 1. They're thinking they're now doing a phase 2-3 combination that they announced they're enrolling this year. So I should update the slide and move that one up. So other projects have advanced to phase 2. In here, Alolis is now licensed to AstraZeneca. I'm not exactly sure where that project is presently, but it's still advancing to my best of my knowledge. So hopefully it will continue. I'll have to check my public releases to see if I see any updates. So that's really the program itself. So what if you're not ready for BPN? If you're not ready for BPN, as I said, DTR has a number of suite things. So if you're in neuroscience space, there are programs I'm going to tell you about, NANDS programs. So not all institutes have these programs, but I recommend you reach out to the science officers you saw listed in BPN in the institute that you're working with and ask them about their programs. They will probably tell you what additional information they have. But I can tell you, NINDS has a suite of programs that is aligned with doing, getting you the preliminary data. So developing an assay and running a screen, or developing a model, an in vivo model for testing, or doing, let's say you have a tool compound already in hand and you want to do the in vivo efficacy study, so you can scale. This can give you the ability to scale that material up, characterize it, and then do your in vivo. So scale it up, do PK studies, show that it's got good things. These are milestone-driven R61, R33 mechanisms, and then run the efficacy study. So typically, no more than 3 years, no more than $499 a year, no more than $750 in direct cost for the whole project. The next receipt date for this is in February. I'll put a plug in. I believe the registration just closed for the 2024 course, but we have a training in neurotherapeutics development, discovery and development course for academic scientists out there. It's a great course. It's here's the website from UC Davis, and the program contact is Arisa. And what's great about this is not only can you go to this and learn about drug discovery, all the course materials, all the lectures from the prior year are up available on this website. So if you just need a primer in PK, it's available. You need a primer in tox, or you want to learn more about intellectual property, there are short hour lectures that are up on these websites so you can learn more about these different areas. It will help you in writing your own grant and figuring out what's important for BPN. So some things to do. I tell this to everybody on every webinar, every time we talk, read funding opportunities carefully. Each funding opportunity is its own thing. It has different requirements, and you need to make sure you follow the requirements to do what it says it's asking you to do in order to make sure your application is accepted, not withdrawn. You do want to make sure you discuss your proposal with NIH staff if you can. I talk to people all the time, and Rakonda manages my calendar, and people make email requests. Rakonda gets them on there very quickly, and we can talk for 30 minutes about your particular program, what's happening with it, what you want to do, what kind of milestones you're thinking about. I can help tell you where I think you fit best in this, and give you some pointers on how to utilize our website to get you the information you need to make a competitive grant. You also get an opportunity to talk to the Home Institute's program director about the disease interest. Contact the SBIR program director if you have SBIR specific questions. Make sure you stick to the page limits. There's only a 12-page limit for the research strategy. It's not a lot of pages. You cannot circumvent it by using really tiny font or really tiny spacing. If you do, the people that receive referral will sometimes withdraw your application. I'll never see it. And there's no way to appeal it if you've gone around their rules. You just have to wait till the next receipt date. Make sure you put forth a solid scientific premise based on preliminary data from the literature as well as your own, and make sure you address the rigor of that data in your research strategy section. Do not bury the rigor discussion in the vertebrate animal section. You need to discuss why the data is rigorous. It's blinded, it's well controlled, it's properly powered, all those good things to say that the conclusions you're making are based on a solid foundation. Make sure you address the obvious criticisms of the chemistry and biology. So many times in review, I see this happen where someone assumes that it's obvious that we're going to fix X, Y, Z in the molecule, but they don't say anything that they're going to, and then they get dinged because their molecule has a known flag or structure alert in it, or has a known off-target effect. And if it's not discussed, if you don't say it, you didn't write it down, the assumption is you're not going to do it, and therefore you're going to get a poor review because of that. Consider contacting a medicinal chemist or BPN staff if you're not sure what those liabilities are. We're happy to look at them and point out sort of the obvious stuff to you. Clearly indicate what will be done as part of the grant and what is expected to be done in BPN contracts. The funding opportunity calls this out and requests a table. Every grant cycle, I get grants that don't have it, and again, it confuses reviewers and makes it harder for them to review your application successfully. It makes it harder for us, even if it is, to figure out what your budget should be. And so make sure you do include budget sheets for all years of the grant. Again, really, really important to do. If you don't do that, it makes it extremely difficult for us to get your application through the review cycle. Some other things to do, make sure you have that preliminary data, address the rigor, and now you have to have a data management sharing plan going forward. Make sure you have included that. Consider if you don't have your registrations in place, you need to doing this 6 to 8 weeks in advance. So if you're coming to the February receipt date, today is a good day to register yourself in these different systems that are required to do this. If you go to the SBIR site, there are 5 different registrations that you have to do. If you're a first time registrant, make sure you got your registrations done, because if you don't, you can't submit your application. And the system, again, there's not really anything I can do at that point. Consider submitting your application early, and early can be a day or two early, because that gives me an opportunity to look at your application and see if, I see you've forgotten to put in something. I can say, did you forget this? And you have an opportunity to fix it before the deadline. For a while during COVID, we were having a longer, a bigger grace period, but those periods are over, and we're back to the standard late submission policy, which is we don't grant any late submissions in advance. You have to meet the policy description, and somebody in receipt referral will make a determination whether they'll accept it. It's not a programmatic decision on our side. If you have any budget year in a UG3 program that's above 500K, you need to ask for permission to submit the grant in advance. Again, 6 to 8 weeks in advance. Let me know, because it takes a while to go through the process. And depending on which IC it is, it may have to go through more than one process. It might have to go through their process and our process at some point. So also talk to your tech transfer groups and your business development groups about your grant application, how you plan on funding the patents, because again, you can't put patent funding as part of the grant application. Talk about licensing activities, make sure you have your intellectual property figured out in terms of how you're going to share it with each other. Some things not to do. Do not plan for this to be your sole funding for your lab, because it's milestone driven. It can and does end abruptly at times. Do not under-resource your budget in order to avoid the limits. If you really need that money, you should be talking to me about the budget 6 to 8 weeks in advance and justifying it. If it's well justified, we have accepted ARAs on occasion for programs to go over that budget limit. Sometimes it's a matter of balancing what we're going to do through our contracts versus what we're going to do in the grant to make that work. Do not plan for the BPN contracts to do disease biology. I mentioned that's not supported by us, or one-off in vitro things. I would also recommend, if you have a good access to rodent PK, that you budget that yourself if you can within your budget, because you'll have better ability to run them faster than the government contracting world. But we do all species of PK. Do not code your application as a clinical trial if in fact you plan to use our BPN contractors to do the clinical trial. Again, I get applications every round about this. And you would, if you're going to have a clinical trial that you're funding within the grant budget itself, it most likely will be a delayed onset clinical trial, because you do not know all the things that are going into it at this early stage. But in most cases, it's going to be done through our contracts and healthy human subjects, and all the things you would put in an application that are required for human subjects, we have to do as part of the contracting process later. So that's why it's not here. There is a program, as I mentioned, for both small molecules and biologics. The biologics group has funding opportunities that have a final receipt date in February. I suspect they'll go through and continue, but presently this is the final receipt date. And if in fact they are renewed, they'll have receipt dates that match us going forward, as they had in the past 3 years. Chris Bosch Office of the Contact. It covers antibodies, peptides, proteins, gene-based therapies, cell therapies, and other emerging biotechnologies. It's identically set up. The contracts are supported differently in that, because it has all these different modalities. the contracts have a little more flexibility to them. And additionally, they have a little more flexibility in their time. They can go up to 2 years for UG3, but still it's a 5-year maximum. We have another program if you happen to be doing pain therapy development, pain therapist development. This program is modeled after the BPN. It takes both small molecules and biologics. It is an RFA, which means it has a funding set aside. So it doesn't have the, if you're in pain area, it doesn't have the budget limits that I talked about before. But it has, you know, a 5-year maximum. You do have to put in how you're going to deal with a pharmacodynamic biomarker here. If you can access one, there's an expectation that you will try to do that within the program. Marianne Pellemounter is the program director for this program, and I encourage you, if you're in the pain area, to reach out to them. This takes every pain indication and takes you all the way through to the phase 1, just like BPN. So with that, I'm going to move it over to questions. Questions can be on application process, review process, proposals, contracts, intellectual property, all of that. So, Carolyn?
Yes, Chuck, we have a lot of questions and we have posted them in the other application.
Okay, so I saw at the top or the bottom?
The top.
Okay.
The first,
so the first question talks about the deadlines are July and August. What are the target dates for funding start dates? So the February receipt date goes to October Council, which means that would be funded probably by the end of the year, somewhere around the December timeframe. The July receipt date goes to the January Council, February Council, can be funded somewhere around March, April. So they're not exactly aligned because these are every 6 months and the council rounds are every 4 months. If you want to read me them, it might
Sure.
be easier.
Yeah, sure. What are the grant mechanisms that support assay development and screening?
If you're within NINDS's mission, that would be in Ignite. The exact mechanism I'd have to look up, but it's going to be in the webinar slides that you have here today.
I'll place them in the chat as well.
Okay, next question. Could 505(b)(2) products with repurposed drug entering the program at the IND enabling tox study phase make a competitive application?
It's a bit of unknown. It's a difficult thing because repurposing that in a generic sense, you would really have to say how you're going to make a commercial product out of this. It's not going to be immediately sort of stepped on by all the other generic makers out there. You know, so if you can articulate a path to commercial viability that makes sense, it's potentially acceptable to come into the program. But you really do have to make a good case because it's going to be a lot of pushback on whether you can invest in this down the road. And as I said, the whole purpose of the program is to get someone to pick it up and take it further into registration.
Okay, next question. Can we develop an assay in discovery phase?
You'd have to have some semblance of an assay. We have had some people develop assays in the discovery phase. Like I said, if you can do it within 6 months and be up and running, then yes, I would say you could do it. Like for instance, I've had some people who come in with ELISA-based, assay that's not really able to do the throughput, and we've been able to change platforms, change the way it's being done, and still generate the program. They've run a screen, and most people have run a screen already have an assay that's up and running. So the people that are the boat that you just described, you typically people that have run phenotypic screens. In those type of cases, again, you have to make the case for the phenotypic and how you're going to optimize the molecules. And so if those assays are something you can put in place and will have sufficient people to try the program, then you can.
Okay, next question. Does IP free of obvious roadblocks require a provisional on file, or is that determined by IP counsel with a patent lawyer?
It'd be more of the latter. Provisional application in many cases is not, well, it's not expected in early stage programs. And sometimes it's more hindrance than help because it creates prior art. And if it's not well laid out, we end up either having to abandon it, or if it publishes prior to that, then it becomes prior art against you. You have to work around it to get IP that's actually protects your hopefully candidate down the road. But what we're really looking for is some idea of that you've done patent searches, that there's freedom to operate in the area you're talking about. This comes up most often when people are especially working in highly competitive areas or areas for which there's a lot of other similar type projects, different ion channels and kinases. How are you going to develop the pharmacophore that is novelty?
Okay, next question. The budget limit mentioned, the budget limit mentioned is a direct cost. Can we request additional indirect costs as a small business?
Okay, so under the two programs, I'll have it. Yes, the answer is yes to both. Indirect costs, you negotiate a rate with the government. Under SBIR program, you also have the ability to charge a fee, which you cannot do under the UH3 program. So not only can you do indirect costs in the SBIR program, but you also have the ability, because it's an SBIR program, to have a fee.
All right, next question. For contracts directly paid, is there a list of estimated costs for service from the CROs to help us put together a budget for submission, or would we need to reach out to get quotes from them?
If you're utilizing this is a really important point. I'm glad you asked this question. I did not make it clear enough. Those budgets are outside of your grant budget. So the contracts you don't need to have any budget for. We'll handle the budget for, completely separate from the grant budget. We'll negotiate the costs and pay the costs separately for it. You need to know that we have the capability to do what you're asking us to do. So if you're doing something exotic, it helps to talk to us about it to see whether we actually have the capability to do it, or we're going to ask you to do that yourself. And also just making sure that you balance it. But keep in mind, in the grant application, it does request you have a table that clearly says, here's what I'm doing, here's what I'm requesting the Blueprint to do for me through their contract or through their consultant. And that way it's clear. And so everything in the review point of view, they say, this is what they say they're going to do. Here's the budget that goes for what they say they're going to do. The stuff over here that you're asking NIH to do, not only is it budgeted by NIH separately, it's also not subject to review. So you don't have a conversation as, is this the right plan? There's stuff for toxicology. We know it'll be done according to industry standards.
Okay, next question. Are there full application examples available as it happens for SBIR, STTR?
I wish there was. There are not. Unfortunately, applications are strictly confidential and are never published. So I cannot put up someone else's application for instance for someone to see. I would love, there's some great applications. I've always encouraged people to reach out to. You do have a list of, people who funded through NIH Reporter, as well as on our website for who's funded for BPN. Then you can certainly reach out to people and ask for any advice you see they can give you, but there are no applications.
Okay, next question. What application avenues exist for CROs, CDMOs looking to participate in the BPN or similar programs?
Okay, so I assume this is asking about becoming on our list of CROs that we've contracted with. The contracts come up for renewal periodically, and they're bid out through sam.gov, like all government contracting. Most of our contracts are IDIQ, indefinite delivery, indefinite quantity, meaning that we qualify a number of vendors up front, and those vendors then would bid on any project work that happens over the period of the contract. So first things first is get yourself registered on sam.gov, and as a government contractor, and then place your bids on contracts as they become available.
All right, next question. Does the budget and the submission cover only the grant, or does it need to estimate the external contractor cooperative work?
Yeah, I answered that question already, but no,
Oh,
it does not. It does not require you to cover the work that's done through NIH contractors. If you're contracting it yourself, for instance, you're doing outsourcing your disease SAR, disease in vitro assay, for instance, to XYZ vendor, that would be in your budget to do, because we don't do disease biology in the NIH CROs. But if you're doing, and for instance, you may choose to do rodent PK yourselves, and you could budget that within your budget, and then you would control those PK studies and contracting all the work associated with doing that work yourself. And you could leave the other contracting stuff for like large animal PK and tox and stuff to NIH. You can do a mix of things like that. But things that were done through our contract, we do it. And things done through yours, we do.
next question. Can we include legal expenses in the budget?
No.
Someone asked if there's going to be a similar webinar for the BPN Biologics Blueprint Grant.
Yes.
Okay.
Yeah. Yes, there will be. If it hasn't already happened, like this one, it will be recorded. So if it's already happened, then you could watch the recording. You could reach out to Chris Boshoff. He'll tell you when the next one is.
I'll place a link to the recording in the chat.
Okay.
Okay, next question. What is the typical capacity and timing of medchem and PK tox capabilities through the NIH contracts? Number of compounds per cycle and cycle time, example, weekly, biweekly, monthly, etc.
Sure. So if you look on that milestone thing I mentioned, the frequently asked questions, the presentation there has all the resources by stage on the right-hand side, but in the typical amounts. But in exploratory, we typically have an effort of around 1.5 to 2 chemists for an 8 to 10 month period doing benchmarking work. When we get to lead optimization, hit the lead, we're typically around 4 chemists. The number of compounds per week is strictly based on the difficulty of the chemistry involved. How amenable is your project to analoging? So, excuse me, I've had some projects that are natural products, very difficult to analog. And I've had others that are quite easy, and it'll be 20 week, 30 week, and some that'll be 4 or 5 week. So it really does depend on your project.
That's all we have right now. We can give it a couple more minutes. If any more questions, please put them in the chat section below. And for those that did join later, we will be sending a copy of the presentation along with the PDF slides in a couple of days.
Again, thank you very much for taking the time to listen to the seminar and learn more about BPN program. I hope it's helpful. Hope it's interesting. Program staff are here to help. Here to help answer your questions. Please reach out and enjoy the rest of your day, week, and the holidays to come.
Thank you, everyone. Have a good day.